Okay, so good afternoon. My name is Aya Zami. I'm a PhD student at Professor Itai's lab at the Israeli Faculty of Medicine, Tehran. And today's talk is about exploring the impact of suboptimal condition on cohesion regulation and chromosome organization. Starting with a previous study of Pilbel and his colleagues, a chromosomal duplication as a transit or usually solution to stress that was published in 2012. It showed that environmental stress can trigger chromosomal duplications in yeast that leads to aneuploidy as a first-line defense, and which is an important concept for understanding stress-induced genome instability. So moving to the cohesin complex, cohesin is a conserved ring-shaped complex that involved in maintaining genome stability. It is composed of 5 key subunits: SMC1, SMC3, SSC1, SSC3, and BDS5 regulated cohesin. It is also involved in 3 major functions. The first one is holding sister chromatids holding sister chromatids together after DNA duplication, and it ensures accurate chromosome segregation. It also involved in regulating the chromatin groups and regulating gene expression, and it also supports DNA repair. So cohesin complex is well studied and well known under optimal condition, but its behavior is barely studied under stress. So we thought that if cohesin misregulates under stress, it might contribute to the aneuploidy, which is the highlighted phenotype that I mentioned in the previous study. Moving to the results, my experiment outline was growing yeast to the mid-log phase, then separated them to 2 flasks. One flask where yeast were grown in the control condition, which was the 30 degrees, and the other flask where yeast were grown in the 37 degree, the under heat stress. And then I have done the extraction and proceed to experiment. My first extraction was the RNA level. As we can see here, when yeast were grown under heat stress, we detected almost a 50% reduction on all the cohesin subunits. However, in the protein level, we detected an alternate reduction of cohesin subunits. The subunits that were reduced were the MCD1, SMC3, and BDS5. And following this reduction in the BDS5 level, we decided to continue and to study the BDS5 subunit in a more detailed way. So cohesin is a regulator subunit of cohesin. It helps maintaining sister chromatid cohesion by stabilizing cohesin on DNA. It supports cohesin loading and establishing during S phase. It works with WAPL1 to regulate cohesin release when needed, and it also ensures chromosome structure and genome stability under stress. So, following the reduction in the BDS5 that I have shown previously, we expected that this reduction may lead to a chromosome to be unstable or destabilizes cohesin. So as expected, our cheap qPCR data showed that when stress were grown under heat stress in red, we detected a reduction in the occupancy of the BDS5 subunit at regions where cohesin is normally enriched, the centromeres 3 and 4, the rDNA locus, and the CARC1, the arm region. So, following the reduction in the stability of the cohesin, we anticipate this reduction. We anticipated a reduction also in the SMC3 acetylation. SMC3 acetylation is a key modification that is added by ECO1 during S phase, and it's important for stabilizing cohesin on DNA, and it's essential for proper sister chromatid cohesion. So we detected a 30% reduction in cells that were grown under heat stress by Western blot, and we can see that here in the band intensity. So such a reduction in the cohesin stability explains why aneuploidy increases, which means that weakened cohesion leading chromosome to missegregate. So, following the reduction in the cohesin binding and the SMC3 acetylation, we hypothesized that this reduced may defect the global chromatin compaction. So, we had 3 biological reports for the chromatin organization. The H2B M cherry is for the block chromatin, the net1GFP for the rDNA loop, and the W for the total chromosome. And as we can see here, when yeast were grown under a heat stress condition, 37 degree, we could see clearly more diffused and decondensed signals, and in the quantification graph here, we can see that also when cells exposed to heat stress, an enlarged chromatin area. So next we ask whether the BDS5 will change its interaction partners under heat stress, and for that we use the Turbo ID system. Turbo ID system biotinolates proteins that are proximity close to the BDS5 subunit, and allowing us to identify both stable and transit interaction. And in this approach, we revealed a Psych8 as an independent temperature interactor. It showed a threefold increase in labeling under heat stress compared to the control condition. So Psych8 is a transcriptional co-repressor that partners with the one to regulate gene expression by repressing or modulating stress responsive and metabolic genes. So until now, there was no evidence about a direct interaction between a Psych8 and BDS5, and we successfully identified this interaction using the IP assay, as you can see here in the Western plot here. So next, we suspected that BDS5 might contribute with Psych8 in regulating the Psych8 phenotypes, and we hypothesized whether the depleting of the BDS5 using the auxin-inducible system would be sufficient to mimic the Psych8 phenotypes, that they are the cell flocculation, the cell wall, and the microtubules. So starting with the first phenotype, as we can see here, here's an auxin is added, means a BDS5 is depleted, and as we can see clearly, we have a strong flocculation, a more clustered cell, contrary to the undepleted BDS5, a well-separated cells. And in the Fox plot here, the red is when auxin is added, the BDS5 is depleted. We detected a higher percentage of aggregates compared to the undepleted BDS5 in gray. Moving to the next phenotype of the cell wall thickness, as we can see also clearly in the microscope images, when auxin is added, BDS5 depleted, we detected a thicker cell wall compared to a uniform outline of cell wall when BDS5 is undepleted. And in the box plot here, it shows that when BDS5 depleted, the cell wall length increases. Moving to the third phenotype, Psych8 phenotype, by counting the percentage of the cell, we could see that we have the control condition, the undepleted BDS5. We detected an 85% of cells with a full length of microtubule compared to a 50% of cells with short microtubule. But when BDS5 is depleted, we detected an 80% of full length microtubule compared to an 80% of short length microtubule. So, for the take-home message, changes in chromatin organization are associated with heat stress. Heat stress destabilizes cohesion subunits and lead to reduced BDS5 in chromatin. Psych8 is a newly identified BDS5 interactant that shows differential binding under optimal growth condition versus stress condition. BDS5 depletion affect phenotypes associated with Psych8 regulated genes. And thank you.
So the cohesion itself can, I mean, that's what I mean.
It's a very nice talk. So regarding the effect on the microtubules, what seems to be seen in those slides that you showed is actually different length of the magnetic spindle.
Yeah, exactly.
And so, how do you know that it is not just a cell cycle phenomenon that has accumulation of spindles in different lengths and not direct effect on the microtubule?
As much as I know about the Psych8, like interacting gene, it regulates the genes that are involved in the spindle length. So
you don't have BDS5, so you don't have cohesion. The cells will rest during mitosis.
So this interaction between Psych8 and BDS5 and how does this like affect the spindle, and that is we are studying that like in this period, and so maybe we'll know that in our next.
Thank you.
It's good that Leah asked that question because next on the podium is Leah's student, Mayan, and I just noticed that I'm Maya and Aya and is changing her place to Mayan, so we all have Aya in our name. We do this.
Aya, Maya.
Aya, Maya, and Aya. Anybody else with Aya in their name?
Raya?
Raya.
Leah.
Laya.
How do I, זה להזיז? זה קדימה? אוקיי. אוקיי.
So busy with my realization.
Oh my God, this is spoiler. Okay. So thank you for the organizers for giving me the opportunity to present my work. And so I'm Ayan, as mentioned, from the Agedov's lab in the Ben Gurion University of the Negev. And I'm going to present my work, which you're going to see in a minute, that slightly also connects to microtubules, as I mentioned before from the previous presentation. So as we all know, biology is the only science where multiplication and division actually mean the same thing. And by that, of course, I refer to cell division, or more specifically mitosis, which is the process in the heart of our study. We specifically study enzymes that are crucial for mitosis. So we study kinesin motors. Kinesins are molecular motors that utilize the hydrolysis of ATP in their motor domain to bind and walk towards the usually plus ends of a microtubule track. Kinesins have canonical binding sites to microtubules, so this is alpha-beta tubulin, and this is a motor domain of kinesin, and there are several common features between the family members, as binding sites such as loop 12, alpha 4, and so on. Kinesin 5 specifically, which is what we study in our lab, are homotetramers. They have the motor domain at the N-terminus, then there's the central stalk and C-terminus tail. This unique architecture of the kinesin 5 motors allows them to cross-link and slide apart antiparallel microtubules, thus making them so important during mitosis for spindle pole separation and proper chromosome segregation. Now, up until now, I said that kinesins are plus end directed, moving towards the plus ends of microtubules. But actually, some fungi kinesin 5, such as SYN8 and KIP1, which are expressed natively in Saccharomyces cerevisiae, were found to change directionality, to switch directionality, but the molecular mechanism of this process is still not completely known, although the biological importance was already suggested for this motor. So we want to find this bidirectional stepping mechanism of these motors. When we look at the amino acid sequence elements of several kinesins, what we can see is the kinesins that were found to be able to switch directionality have a long N-terminal non-motor domain extension. The motor domain only starts here. That does not exist in exclusively plus N-directed kinesin 5s and kinesin 1. So this led us to believe that this region might have an effect on the bidirectional stepping mechanism of these motors. So in a previous study we published just last year, we found in cryo-EM a low electron density. This is the motor and these are the microtubules. We saw a low electron density coming very close to the microtubules, and in collaboration with Itamar Kass, we performed molecular dynamic simulations, and we found that this region of the N-terminus indeed likely interacts with the C-terminal tail of beta tubulin. So this is what we understood from this study. I will say that the cryo-EM was only performed in the presence of AMP-PNP and ATP analog. However, we do want to understand the mechanism of these motors. So we want to understand how exactly this region rearranged during the ATPase catalytic cycle. So to find this out, we decided to utilize a very important tool, FRET, fluorescent resonance energy transfer. Just a reminder, we need to have a spectral overlap between the emission of a fluorophore donor and the excitation of the fluorophore acceptor, and they need to be in close proximity until 10 nanometers to be able to transfer the energy. So I can excite directly the donor, and if they're close enough in proximity, I can actually get an emission peak of the acceptor because of energy transfer. So in our case, we labeled the N-terminal non-motor domain with our donor, and the microtubules with the acceptor. More specifically, we used fluorescent Taxol. So the distance between the residue that we labeled on the sienate and the Taxol is approximately 4 nanometers, which is well within the distance required for FRET experiments. We'll just say that also as a control, we utilized, we labeled more further position that's still within the 10 nanometer distance of FRET, but a lot further. Okay. Oh, so this is the position. Okay. So how we do it? We didn't want to use cis-light, variant because it was non-functional for our motors. So we wanted to go with other method. We collaborated with Mira Amiram from the Biotechnology Engineering department in our university, and we incorporated a natural amino acid specifically at the position that I want to label. So we just talked about synthetase as well. So in here we incorporate an orthogonal synthetase and orthogonal tRNA that recognizes what usually is a stop codon and do what we say amber suppression, changes its function. Instead of stop codon, it incorporates the desired unnatural amino acids, so that at the end, yes, we get a protein with an unnatural amino acid desired a specific position. We then fluorescently labeled our protein using click reaction. We have decided to go with strain-promoted click reaction. We also tried before copper catalyzed, but we moved to strain-promoted. So let's look at the process. So we begin with unnatural amino acid incorporation. We incorporate PAZF that has azide. We then continue to the labeling with the donor with a click reaction with DBCO, so it's a strain cyclooctene. And then we do a functionality assay. First, we want to make sure that these proteins, these labeled proteins, can actually bind to microtubules. And then we want to see how it affects the energy transfer between the motors and microtubules. So I did a two-step purification, and already this step we can see that the incorporation of unnatural amino acid worked well. The tag didn't act as a stop codon, but indeed the unnatural amino acid was incorporated, and thus we get the proper size of our protein, which was first step very important. We then continue to the click reaction. So again, this is our unnatural amino acid, and this is the dye that we use. So the click reaction happens here between the azide and the alkyne. So we get labeled amino acid at a very specific point. What we can see here is gel, of course. This is homocysteine, and this is gel fluorescence, but these are the exact same gels. And we see that the click reaction works very well because in the presence of the dye, we can see it in the fluorescence gel. Of course, in the absence of the dye, we cannot see it in the fluorescent gel. So the click reaction also worked very, very well. We can now move to the functionality assay. So microtubule copelting assay is an assay with which we take in vitro polymerized microtubules, combine them with our variants of sienate, centrifuge them to separate the supernatant and the pellet. The microtubules are heavy, so they should be found in the pellet, and free proteins should be found in the supernatant. Since our protein should bind to microtubules, they should be found mostly in the pellet. This is what we expect. This is if they bind properly. So we ran the gels in this case, and I want to just focus on here, where we can see that in the presence of the dye and the presence of microtubules, so this is the microtubule band, this is the sienate band, we can see enrichment of protein in the pellet in the presence of microtubules as opposed to the absence of microtubules, which means there are sienate motors that binds to the microtubules. We can also see it again in the in-gel fluorescence. It's less enrichment, but it's still there. So there are labeled proteins that are bound to the microtubules. Yes. So now moving on to the last result. Actually, this is the FRET assay. We're using again sienate labeled with the donor and labeled Taxol for the acceptor. And at the beginning, we started with the same condition as the cryo-EM, which is AMPPNP. And we can see that the blue curve is in the presence of an acceptor. The orange curve is in the presence of unlabeled microtubules. So we can see that in the presence of an acceptor, there is a major energy transfer to the acceptor. We see an increase in the acceptor emission peak and also a decrease in the donor emission peak. So this does tell us that there is an interaction between these two regions. They are in close proximity. We also wanted to try different stages of the catalytic cycle, as I mentioned before. So we tried ADP, and we also saw here, to our surprise, very nice energy transfer. So not only in AMPP and P, as we saw in the cryo-EM, there is close interaction between these two regions, but also in the presence of a DP. As a control, I remind you, we wanted to label for this position. This is still preliminary data, but with AMPP and P in the first position, we almost don't see any energy transfer, which makes us believe that our assays actually work fine. So these all results support the notion that the N-terminal non-matter domain interact with the microtubules in the presence of ATP and ADP in the catalytic pocket. And if we go back to the bidirectional stepping mechanism, which is our goal to understand this state enables a situation where two heads are tightly bound to the microtubules at the same time. So then the motor can decide which way should I go now. I want to thank you all for listening, thank Lea, and thank Lara that did many of these essays. Thank you very much.
Beautiful to that. I promised Martin to look if he has a question first.
I would like to ask a question. Miracle fermentation. In what circumstances cells need this biodirectionality?
Okay.
What actually do we see?
So why do we need this
Why do we
bidirectional?
need this?
Okay,
Yes.
so specifically, as I mentioned, we see it in fungal cells, in S-POMBECAT7 and kinesin 5 C8 and KIP1 in Saccharomyces cerevisiae. These cells divide via closed mitosis, so there isn't a dynein minus end forward movement that is assisted by dynein. So what we expect is that the motors needs at the beginning of mitosis to walk towards the minus ends, meaning towards the poles, when the poles are still very close together. Then they can capture the microtubules from the other pole and switch directionally to push the poles apart. So this is the importance of this movement.
And nearly completing the set is Ava from Ronit Weissman's lab. You just needed a tiny little tube to put on your pin, you would have joined the...
I would have joined the club.
Exactly.
It's fine. Oh, not Aya.
Yeah.
Almost. Very close. Okay, here you go. Thank you. Hi, so as was just said, my name is Ava. I'm a PhD student under the supervision of Professor Ronit Weissman and Professor Martin Coupier in Tel Aviv University. And today I will be talking about the identification of a novel phosphorylation site in the ATP binding pocket of GAD8. So first let's start with a bit of introduction. TORC2, or target-to-bromide complex 2, is a very well conserved complex in evolution, which has TOR1 as its main catalytic subunit. So I know TORC2, TOR1, numbers are confusing, but here I will be talking a lot about the TOR1 subunit. TOR1 is known to phosphorylate and help activate its downstream kinase GAD8. It's phosphorylated at 2 of the 3 needed phosphorylation sites for the full activation of GAD8, the TOR motif and the hydrophobic motif. The TORC2 GAD8 pathway is known to be important for various things that I will say in just a minute, but the full role of the complex is not well known. But we do know that if we delete either TOR1 or either GAD8, cells cannot survive under stresses. So the TORC2 GAD8 pathway is needed for nitrogen starvation, glucose starvation, genotoxic stress, temperature stress, and more. So we know that, and we are interested in better understanding the regulation of GAD8. So the autolog of GAD8, AKT, if it is hyperregulated in mammalian cells, it leads to cancer. If it is downregulated, it leads to diabetes type 2. Therefore, if we try to better understand the tight regulation of GAD8, we might better understand also its cellular roles. For that, we decided to perform phosphoproteomics experiments, and we identified many different phosphorylation sites within GAD8. Some of them were previously identified, and some of them were novel. Out of the novel sites, the one that was the most of interest to us was the S240 putative phosphorylation site. This site is found within the ATP binding pocket of GAD8. Now, ATP has a negative charge. Phosphorylation also has a negative charge, so this suggests this site to be an inhibitory phosphorylation site. So what did we do? We took GAD8, we took the S240 residue, and we mutated it to either an alanine to create a phosphonol mutant, or to an aspartate to create a phosphomimic. Then, if you remember what I said a minute ago, either deletion of GAD8 or deletion of TOR1 causes the cells to become highly sensitive to stresses. But what happened in our mutants? In the GAD8 S240A mutant, the phosphonol, we can see here that it looked just like the wild type. However, the S240D, the phosphomimic mutant, became partially sensitive to stresses. This fits with the idea that this site might be an inhibitory phosphorylation site. Then we turn to biochemistry. So, first of all, what we will be looking at now is one of the TOR1 dependent phosphorylation site, which is the S546 phosphorylation site, which is a very good marker for TOR1 activation. As you can see here, in wild type, when we are in rich media, we have high level of this phosphorylation, but when we are switching the cells to glucose starvation media, the phosphorylation goes down. Within the time course in the wild type, you can see that within 5 minutes already, we have a dramatic drop in the levels of this phosphorylation. However, we can see here that within 2 hours, the phosphorylation comes back. What happens in our mutant? Indeed, in the S240A mutant, we can see the dramatic drop, and we can see that it returns around the same time as the wild type. However, in the S240D mutant, while it does drop within 5 minutes, it comes back within 10 minutes. This suggests to us 2 things. First of all, we saw that this result was specific to glucose starvation. So in another stress media where the TOR1 dependent phosphorylation is known to go down, this did not happen. So this suggests to us that that the S240 is acting specifically in glucose starvation in this case. And this also suggests to us that there is a feedback loop inhibition between GAD8 and TOR1. So we have TORC2 and TOR1 phosphorylating GAD8 at 2 of its sites. Under glucose starvation, we lose this phosphorylation, and this is dependent on GAD8 activity. However, when I have the S240D mutant, we do not have this feedback loop inhibition, and therefore we keep having high levels of the TOR1 dependent phosphorylations. Thank you.
Sorry, I'm getting great. Thank you for a beautiful talk. And do we have questions? Ava, help me to speak.
Yeah. You know, when cells are growing in cycling conditions without any arrest or stress or other factors, what is the time in the cell cycle in which GAD8 is phosphorylated at that site, at the ATP site?
But within the cell cycle.
Cell cycle of cycling cells.
So I did not check within different cell cycles when that residue is phosphorylated. We do know that in rich media in general it is phosphorylated, But I can tell you, for example, that our assay in glucose starvation, the cells are not yet dividing and growing. So I do know that the return of the S200, the S546 phosphorylation after 2 hours is not dependent on, is not within growth condition, even though the cells are asymptotic.
The reason I'm asking is because your mutants are constitutively stressed, so you may want to be careful regarding the state that it may be effective or not effective when you may do just not change about stress, etc.
So quick question, because you finished early, which is wonderful, that I have.
Yeah.
Do you know if it's also inhibiting tor-mediated phosphorylation of other substrates, or is it kind of a very local inhibition?
So we are searching for, another project in the lab is also searching for GAD8 substrates. And for now, what we found is indeed we have a protein that is regulated by TOR1 and GAD8, and we found that the levels of that protein are definitely dependent, and they are much low. So in TOR1 cells, the levels of that protein are much lower than in the wild type, and the S240D mutant, we got the same results, also showing that indeed it has lower, potentially in vivo activity. So this is also one of our candidates that we identified.
Really nice. Thank you so much for this presentation. Our next speaker is Matan.
And it's also, hi everyone, my name is Matan, and I'm a first year PhD at the Faculty of Agriculture. I'm actually a plant scientist, so I'm using yeast quite differently from what I've heard so far, and some might consider exploitative. So I'm going to talk about my research of how we leverage yeast as a cell factory for metabolic engineering of plant specialized metabolites, specifically a newly discovered isoflavone in wheat called trititine. Plant produce a remarkable diversity of specialized metabolites that mediate ecological interactions and plant defense. Among them, we're going to talk about isoflavones, which are a subgroup of flavonoids predominantly found in the legume family, Kitniot, and mediate plant defense. Their plant defense role makes them quite interesting in agricultural terms as an eco-friendly alternative to chemical pesticides and insecticides. But more interestingly, you can see their structure. Their structure resembles the human hormone, the human estrogen. And thanks to the similarity, they also have several health-promoting properties, as you can see here. And thanks to these properties, they have been co-opted for the pharmaceutical and nutraceutical industries. However, the production of these isoflavones in the industry is only through direct plant extraction from soybeans specifically. So producing them in microbes can offer a clean, scalable, non-agricultural, and eco-friendly alternative for the production of this isoflavone. I told you it's totally different.
We love it, don't apologize.
Yeah. So using the baker's yeast is quite common in metabolic engineering. They are fast, they are scalable, modular, and robust, and also genetically flexible, and especially for reconstructing complex plant pathways. One of the main advantages is that they can efficiently express the membrane-bound cytochrome P450 enzymes, which are crucial for many plant pathways, including isoflavone, and also poses a major bottleneck in other microbes, for example, prokaryotes such as E. coli. So this is one example of stably expressing the isoflavone biosynthesis pathway in yeast, the biosynthesis pathway of soybean. The researchers did multiple stages of optimizing this yeast strain to rewire their carbon flux towards the production of isoflavones, and they did it by several genetic modifications. For example, engineering redox partners for the cytochromes, modifying enzyme localization, substrate trafficking, and also the glucoside forms of these metabolites, which are more stable for us. And despite they were able to achieve quite a relatively high titer, we call it titer, the concentrations of the metabolite, there was still a bottleneck in the leguminous isoflavone synthase, which is the most important enzyme in the process. that had shown suboptimal activity, in converting liquiditigenin, which is the last phase, this is the precursor, to daidzein, which is one of the two isoflavones that soy synthesized. But apparently, as I said earlier, isoflavones are not unique to the legume family, and a recent study from my lab discovered and characterized an alternative pathway producing isoflavone in wheat, producing the all-methylated isoflavone called tritizine that was characterized to have plant defense role and antipathogenic activity in wheat. But most importantly for us, wheat uses a different isoflavone synthase and a totally different reaction to synthesize isoflavone. It's also a cytochrome 71, but it may bypass the long-standing bottleneck of the legume isoflavone synthase in metabolic engineering. So the bottom line is there's a new path for isoflavone metabolic engineering. And this is where I come, and my study harnessed this discovery of the new isoflavone biosynthesis pathway to create our own metabolic engineering chassis of in baker's yeast in Saccharomyces cerevisiae in our lab. And apart from building a yeast strain that synthesizes isoflavone, we would like to further characterize this newly discovered pathway. And by reconstructing the pathway in yeast, we would like to determine whether the new isoflavone synthase exhibits higher catalytic efficiency compared to the legume one. And also we would like to test whether this isoflavone synthase has some flexibility in the substrate it can get, and also the product it can synthesize substrate flexibility, or some might call it enzyme promiscuity. So for our genetic engineering, we use quite cool tool called a multiplex yeast toolkit. This is a new tool that allows us to assemble multi-gene constructs in a modular way, which is quite effective for introducing long metabolic pathways. It's a Golden Gate based assembly, starting from the level 1 assembly of the transcriptional units, as some of you know. At the level 2 phase, we can combine up to 10 transcriptional units into one single cassette. And once assembled, it can be integrated into the yeast genome in defined loci using CRISPR-Cas9 mediated genomic integration, homologous recombination in the case of yeast. So this is where we stand now. Oh my God. This is where we stand now. The first stage is to establish a yeast strain with the biosynthesis pathway of tritizine, which means a yeast strain that expresses all the 11 basic genes that are responsible to synthesize tritizine. You can see the pathway here, while 4 of the genes belongs to the general phenylpropanoid pathway, which is the pathway that synthesizes, among other, flavonoids. They are we took it from Arabidopsis, and they are quite conserved throughout the plant kingdom. And the branch of the tritizine, another 7 genes from wheat. And we took all these 11 genes and we divided them into 3 cassettes and started that targets different loci, and started doing the transformation gradually by piling locus after locus after locus, but starting with the downstream genes that in order to not affect or not interfere the yeast growth, because they just can live there, express, but have no precursor. And then the last transformation was of the upstream genes that starting from the phenylalanine flux of the yeast. This is where we stand now. I'm sorry I'm showing you PCR because I'm still in my first year, but you can see, I'm sorry a lot, sorry. So this is the level 2 cassettes. It took a while, the cloning was long. You can see a cassette for each locus, and then you can see in the last strain these 3 cassettes are integrated into the yeast genome. You can see from the 5' and 3'. And now that we have this desired yeast strain, our next step is to evaluate it, and we evaluate it by measuring the tidal concentration of the metabolite. We're doing it in analytical chemistry by LCMS validation, and then we will decide for further genetic modification that mainly focus on yeast endogenous genes by keeping the trade-off between not interfering in the yeast growth, but maximum yield of our metabolite. Another thing that we would like to do, it's to test its substrate flexibility, so we can do it by genetic approach, introducing other enzymes for other precursor, or easier, in yeast we can just feed them with other precursor and see what we can get. And a bonus that we might do is to test whether this isoflavone from wheat has the same health promoting properties that we see in soy. And if we're going to do it, it will be, of course, in collaboration with other labs. So I'd like to thank my PI, Guy Kulturak, and my lab, and thank you for listening, for opportunity to be here.
Thank you. It's really cool to see kind of the diversity of things that one can do with these. So open for questions.
Yeah.
Kobe.
Yeah, sure. Are you introducing any mutations into your yeast strain to, say, increase the amount of phenylalanine available?
Yeah, so we thought about doing it ourselves, but now it's more reasonable to search for optimized yeast strains. In my case, for several components in the pathway, for example, the phenylalanine flax. Also, we use a lot of malonyl-CoA, heme groups for the cytochromes, and there are, and we just need to get them. It would be easier than that we can focus on specifically on my pathway and not things that people have done already. Thank you very much.
Dudi will catch.
Yeah, exactly.
And they.
It's good. Talk in the lunch break. Good. So it's my great pleasure to now invite Judy Berman from Tel Aviv University to tell us about actual pathogenic yeast that actually do something, not just beer and bread.
First of all, it's an incredible pleasure to be here and to see so many friendly faces, and this is one of my favorite meetings, even before I was a PI here. I've been in Israel for almost 15 years now, after 25 years in Minnesota, and before that in Israel and in the States. I'm going to tell you about a yeast that is in your body, okay, that you should be aware of, all right? And it's in all of us, okay? And I'm going to talk only about work that has been done in my lab here, okay? And basically Candida albicans, the organism I'm going to talk about, has now become finally noticed by the CDC and the World Health Organization as a serious problem. It's in all of us, and it's commensal, and most of the time it's just fine, okay? But if you look at different fungal diseases and their frequency, okay, and they are generally less frequent than, for example, malaria and TB, but they're still up there. When you look at the mortality associated with those diseases, the mortality starts to jump above malaria and TB, okay? And over here is candidiasis, which is the systemic infection with the same organism that used to be just happily growing in your gut. And so it's either a bloodstream infection, or it's a deep-seated within the peritoneal cavity, or it can also infect basically all of your organs. And so that's what we're going to talk about. And the big issue is treatment failure is incredibly high. Okay, about 30% for systemic Candida albicans infections. For some of the other species, which are becoming more prevalent because they're more resistant, it's much higher. Okay, so what contributes to this treatment failure? Three major things. One is immune system. A lot of the people who are sick with this have immune problems, and therefore Candida becomes much more of an issue because our immune system normally keeps it in check. It can also be because drugs used to treat it can't get to the disease. For example, if it's infecting the peritoneal cavity, There are wonderful drugs that can do something, but they can't get in there. Okay, and the third issue, which I'm going to focus on because I'm a yeast biologist and once upon a time I worked on Saccharomyces, is the properties of the fungus itself, drug resistance, and drug tolerance. And I'm going to talk about drug tolerance. This name was forced upon me. I wanted to call it perseverance, but I now believe that resilience is a perfect name for it. At the same time, tolerance is what we've been using for quite a while. It's confusing because it's not the same as bacterial antimicrobial tolerance, but I'm going to explain what it is, okay? Candida albicans is the most common cause of candidemia and invasive candidiasis, okay, because it's already in a very convenient place to cause it. Now, where are we talking about? You are, wait a minute, you are here, okay, Cerevisiae. Candida glabrata, if you want to work on something more like Cerevisiae, take glabrata. It's haploid, okay? But it doesn't have on its own a sexual cycle, but it can be kind of engineered almost. And we are here at the top, okay,
This chart.
of this chart. It's a common commensal in pretty much all of us. It's easily detectable in about 85% of us. If someone wants to check if you have a Candida infection and do some standard test at the local pharmacy, they'll find Candida and they'll tell you you have it and they'll tell you to eat all sorts of diets, but we can discuss the relevance of that afterwards, but pretty much all of you will test positive. It's very prevalent. Mortality is around 30%. So what distinguishes this resistance from tolerance? You are all familiar, I hope, with the disc diffusion assay. You put a disc on the plate. Normally a nice susceptible strain gives you a zone of inhibition. It's fairly clear. If a strain is resistant, it grows much closer to the disc. The minimal inhibitory concentration, or MIC, is inversely proportional to the distance, right? The further away it is, the more susceptible it is. Okay? Tolerance is when you grow this disc for another day and it looks like that. Okay, so it was a susceptible strain. In the clinic, they marked it as susceptible, and the next day it grows like this. Now, I don't know about you, but as a biologist, I'm a little worried about those guys growing in the middle, and that's what sort of drove the study that I'm going to tell you about. This is doing the same experiment with e-test strip assays. They just are commercially available, and it's like they have a little ruler on them that tells you what the MIC is. And here are the instructions for the clinic. Ignore the lawn of microcolonies and read at 80% inhibition, okay? So I don't know about you, but I wouldn't want that guy being treated with that drug, okay? So there's a range of tolerance levels in our strains. Each of you has one of these, and it could be from very intolerant to very tolerant. This might be really worth knowing before we give you a drug. And the level of tolerance is pretty reproducible if you take the same strain and do it again. Okay, so if you take from the middle of the halo and you put it again, you get the same rough thing growing after 48 hours. This suggests that genetic background is really, really important for the diversity of this response. Right? Different genetic backgrounds are giving different reproducible results. Okay, so does fluconazole tolerance actually affect clinical outcomes? We've done 2 very small scale studies. More bigger ones need to be done. Here's one study with strains that we received from Brazil, where they had tested patients who had systemic infections, and the MIC of those the MIC, here's the MIC of those strains was about the same when the infection was easily cleared or when it wasn't cleared and it had to be treated with another drug. Okay? But when you look at the tolerance levels at the beginning of the experiment, here are susceptible strains. Here are the ones that were easily cleared, and here are the ones that cause persistent infections. Okay? It's a small study, but it was significant. In addition, here in Israel, together with Ronen Ben-Ami at Ichilov Hospital, that was done with Arnaldo Colombo, who's in Brazil, and Jules Eni. We looked at retrospectively at patients who had been treated with fluconazole and found that if their tolerance level was higher than 50% of the growth inside the halo relative to growth outside, that the mortality was significant, whereas if the tolerance level was less than 50%, none of them had died within the time frame of the study. And this was all based on if they had a diagnosis and started treatment within 24 hours, because another thing is that if you don't treat it fast, forget it. Okay, it's really bad. Okay, so high tolerance appears to drive persistent infections and higher mortality. All right, so what distinguishes antifungal tolerance from resistance? Not all cells form colonies inside that halo. You saw that some of them are kind of light. They also grow slower, of course, they show up slower, and a lot of that has to do, so not all of the cells form colonies. That tells us that there are cells doing something and cells not doing something at the same time in the same population, and they're genetically identical because I told you that you can take cells from the middle and you'll get the same thing again. All right? We did only one single cell RNA-seq, but we see two different subpopulations. So now I'm going to start the ties to all of the people who talked here. One of those is enriched for ribosomal DNA, and the other one is the stress response. Okay? You hearing the familiarity with this idea? Okay, so tolerance is a type of phenotypic heterogeneity where cells are expressing different things. Whether these are always the same or whether they constantly shift between those states, we don't know. The time of colony appearance is delayed. The reason they show up at 48 hours isn't so much because they grow slower, it's because they do something to sort of wait until they become detectable colonies. And we're really interested in what that is. So it's a type of phenotypic heterogeneity that involves also how fast you grow, and the higher the tolerance, the faster they show up, or the shorter their lag time. Okay, and as I told you, it's heritable. The level of phenotypic heterogeneity is heritable. Okay, so we have genetic and non-genetic contributions here. Finally, it's largely concentration independent. If you put more drug in the disc, you get a bigger halo. That's the MIC. That's resistance, right? And it's drug concentration dependent. But note that the amount of growth inside the halo is not very different between low and high amounts of drug. Okay, what this is telling us is that tolerance must be relying on resilience mechanisms that are independent of the drug-target interactions. And just to tie to the last talk, the drug target is a P450 cytochrome, whatever. Anyway, we've done a lot of experiments with inhibitors and things like that. We know lots of things that contribute, and if you knock them out, no tolerance. So all of these different pathways like TOR-C, where's my TOR person? Okay. Protein kinase C, heat shock response, another one of the talks here. Calmodulin and its regulation of calcineurin, and the expression of stress and non-stress proteins. TOR pathway, super important. And all of these are necessary, but none of them are sufficient for having tolerance, okay? So the working hypothesis is that tolerance is due to stress resilience networks, and subtle differences between genetic background must modulate those very complex networks. Want me to say that again? Okay. Yes? Tolerance is due to a bunch of stress resilience networks that make up lots of the things that you guys study, okay, that all talk to each other, all right? And the subtle differences in the genetic backgrounds of different strains modulate how those networks do their thing. Okay, that's my take-home message. I can't tell you all about all the molecular interactions, so every time I try and work on, submit something like this, I get told that we have no mechanism whatsoever and it's not interesting. But I actually think it's incredibly important to realize that, folks, resilience requires lots of different ways of being. You just heard about 4 or 5 or 6 hormones that are all affecting us and our stress levels, right? So why should yeast be any different? Okay? All right, so how do you study something like this? That's the problem. And the way that we decided to go about this was to get involved in a very big study with an ERC Synergy Award that I got a number of years ago with Marcus Rosser, my partner in crime, who's an expert especially in high throughput and proteomics work. So we want to understand multiple genetic backgrounds, the stress pathways, and also what's different between those different cells. This is huge. Okay, so the way that we went about this was by focusing on the, unraveling this complexity at the species, the isolate, and the single cell level. And I'm only going to talk about the species level today. The rest of, all of it's still in process. I'm going to also tell you I decided not to stress out about the fact that I don't have all the final results. Okay? So, how do different genetic backgrounds affect tolerance? So what did we do? One of my strengths is I know everyone in the Canada community, and I can get them to give me their strengths. And they did. Okay? So we got strains from across the world, every continent except Antarctica. We have them mostly from humans, but also from the environment, from plants, from animals. Okay? We have them starting from 1936 with a type strain, all the way up to 2022. Before and after the AIDS epidemic, because AIDS patient presented with Candida albicans on their thrush in their mouths. Okay, this work was a huge collaboration. These are 3 of the people who really focused on phenotyping the things. Some of you may know Nir Cohen, who was an amazing student in Maya's lab and is now in Marcus's lab, and Austin Matola was a postdoc in my lab, and then a postdoc in my lab but in Germany, and now a postdoc in Germany but working with me. Okay, so very collaborative. All right. And what we did was we collected these things. We isolated them for single colonies, found out that, I don't know, like 3 or 5% of them were not albicans at all. Okay. Arrayed them, sequenced them, and we've done phenotyping on them. They've also done proteomics on all of them at one condition, and Austin is doing proteomics on 12 phenotypes across 90 strains, and Nir did high throughput microscopy on all of them. Although I don't think any of us have sat down to try and figure out what that means yet. Okay, so we've done phenotyping, and first of all, the DNA sequence gave us 24 clades of Candida albicans. It's a little more than they used to have, but they're pretty much the same. The little 3 over here were all called one clade before, but there are some significant differences between them that you guys certainly don't care about. There's one clade over there that is like considered to be, by some, a different species, but it's not. They come from all different places. Interestingly, a few of the clades are more enriched in animal samples or plant samples or food rotting things and so forth, but it's a slight enrichment. You can see it on the different colors on the little wheels around there. Okay. Ploidy. I love aneuploidy. I'm not going to talk about that today, but if you put these strains under a lot of drug stress, they can get transient aneuploidies that also make them tolerant. Again, speaking to the fact that many pathways are involved, and if you up something a little bit, you start to get shifts in your pathways. Most of them are diploid. That's what Candida albicans is. It does do mating. It does not do meiosis. I can tell you all about the illegitimate things it does all the time and doesn't care. And that you have meanwhile afterwards found out that cerevisiae can do too. Okay, so there are 20% aneuploids in the diploids, and then the aneuploidy goes up a lot when you start to go to higher ploidies. Okay, and we looked at lots and lots of phenotypes, and here's the drug story, which is that while we originally had 2.5% resistant, after you get rid of the non-albicans guys, it's only 1.64% resistant. That cannot explain the 30% mortality rate. And when we look at tolerance, there's lots of tolerance at all different levels. And if you look at 7 different drugs that are used to treat the infections, all of them have tolerance to some degree.
Each drug is a strain?
Each dot is a strain, yeah. Okay. So this can account for a lot of treatment failure. Now, how do we figure out other associations? And most things were, other than some obvious ones, were not that obvious. So we started looking at the distribution of different phenotypes and then comparing distributions to distributions. For example, here, fluconazole tolerance to caffeine. And you don't see anything much here. The purple guys are from the strain collection that everybody uses to sample the diversity of Candida albicans, just to give you a sense of what you get when you look at 20 strains instead of 1,700. Okay, here we're looking at tolerance, fluconazole tolerance, and the major thing that I can say is that these the samples were a little low on tolerance, and the thing that has hit me in the face from all of these analyses, there's a lot of strains with 0 tolerance, and we have to now start analyzing them and comparing them to the other levels of tolerance, which might help us figure out a lot. But that's something that really I got out of looking through a bunch of things so that I could tell you something. So the other thing is that if we look at most phenotypes, high, low, high, whatever, not much interesting. Here, if we look at growth rate relative to fluconazole tolerance, and we look at the slow growers and the fast growers, the slow growers are a little enriched in no tolerance, and maybe that's part of their story. But I think clearly there's no good correlation between slow growth and tolerance. That's not the story. And I get asked that question every time, so now I have a slide for that. And finally, how many isolates make a representative collection? I don't know if 20 is enough, but I'm not quite sure how to pick how many. And what we were sort of dreaming of is if we had 30, we could put 3 copies of it on 196 plate and send it to all our friends. I think that may not work, but we are making the collection available publicly through the EMBO labs as soon as we publish the paper. Okay, finally, we looked at some phenotypes by looking at the extreme phenotypes and seeing what overlaps. Do I still have half a minute or so? Okay. And what we found are some interesting places where you see multiple phenotypes going together. And when we look at that box, one of them here is caffeine and azole resistance. And then we found out in the literature that Maitreya Dunham did a screen that found this exactly the same thing. But we also had looked at these and we found that there are different groups of genes that are more enriched in the guys that are high caffeine, low azole, high azole, low caffeine, and high in both of them. And so that's the kinds of things that we're going to be doing in the future. So the bottom line is tolerance may be important for surviving fungal infections or not. Genetic background is really important and it controls a lot of things. Many stress pathways are necessary and none are sufficient for fluconazole tolerance. There's not just one way to be resilient, and you may need different paths among those resilience pathways to respond best to different stresses or levels of stress. And of course, the antifungal drug tolerance probably is important clinically. So with that, I'm going to thank all my friends. This is from our first Synergy retreat. Marcus Ralser is an amazing collaborator who has been working with me very, very closely on this. And thank you very much for your attention.
Before we start with the question from the audience, I'm sorry, I'm still stressed from what you said about having just 24 hours to start treatment. When do we know to push the doctor
Usually what we're talking about are patients who are in the hospital who have an infection. They didn't respond to antibiotics. They had a surgery. That very often, abdominal surgery is a real big, not abdominal, but GI tract surgery will release things from within the GI tract, and Candida's in there. And then if they're not responding to antibiotics, they'll very often give antifungals, and now they tend to give antifungals prophylactically also for certain certain treatments. So relax. I'm relaxed, and you know, you can't worry about everything all the time. It's just not.
But I can worry about some of the things.
I read in the past that Candida albicans can create biofilm. Is biofilm can also?
Yes. So you'll read in all the literature that Candida albicans makes biofilms, and it does. And the biofilms are a real problem because in the literature they say biofilms are highly resistant to drugs. But if the same strain is not in the biofilm, it's not resistant to drugs. Therefore, by definition, actually, biofilms are hypertolerant. Now, we know that one of those mechanisms for hypertolerance is it secretes lots of extracellular matrix for the cells to sit and stick and adhere and aggregate. And that stuff, which is cell wall stuff mostly, sticks to the drug, and the drug never gets inside. It's like basically like Velcro for the drug. Okay? There's that scotch.
So one last question. Yeah.
So I may have missed that, but the experiment on the 7 drugs. So if one strain is tolerant for one drug, it's very likely to be tolerant for all the others?
No, not necessarily. What we saw was that there was a very good I went through it too fast. I'm sorry. I just wanted to. I had a lot of new data that I was excited about. When it's an azole, they have very similar tolerance levels. But because it's a similar mechanism of action. Actually, when it's a polyene, which actually has sort of the opposite mechanism, so azoles inhibit ergosterol biosynthesis, which is a sterol. The polyenes bind sterols within the membrane from the outside. So if you're not making the sterols, you can't bind that drug doesn't work. Okay, but very often, so they don't have the same mechanism at all. And you see, I showed you one that was not correlated, and the one that was correlated was 2 azoles. Okay, which was like, I didn't need to study 1,700 strains to figure out that azole mechanisms are related to one another.
But did you find the mechanism that gives tolerance to all of them?
No, and very few of them are tolerant to all. But that's actually a really, write that down, Sophia. That's a really interesting idea to just look at if there are rare ones that are tolerant to all, we could look at what they have that's different. And each of these look at what they have is not trivial, but we can do it. And I should actually say that Shira Milo, who was a student in Shai Kovo's lab, thank you, is now working with me on the bioinformatics of this, and she's phenomenal. So, hats off to her for all the bioinformatics.
תודה רבה רבה. I have another surprise for you, but don't worry, this time you don't have to get up and exercise. You know that one big reason to be stressed is if your PI tells you to go and make a lot of strains to assay them, right? So from the maker of the emojis, I bring to you Dean, which will give you an amazing resource to tackle that little piece of stress in your life. And afterwards, we free you to your R&D session.
So hello everyone. As Maya said, I'm Dean Baruch. I'm from Maya Shorden's lab. And in the Shorden lab, we really like yeast libraries. These are a great tool to work with. This is because some great reasons. They are systematic. You can pick and choose which strain you want to tackle, which protein or gene. It's genome-wide. Usually we make it like for the whole genome, and it's very versatile. You can make a lot of experiments with it, and you can use the strains to a lot of assays. But there is a big problem with these yeast libraries. Where do I find all of these? There wasn't one unified resource for finding all of these libraries. So together with Ophir Klein, I wrote a review, a comprehensive review about Yeast libraries. So I collected all of the libraries that people created, only genome-wide and mostly Saccharomyces cerevisiae. And we also created a website called Library Yeast, where you can find all of the libraries available and who has made them, and in the review is also history about yeast libraries and how they advanced functional genomics. And you can find the review also in the site. This review describes about 40 libraries in details, and it will be regularly updated in the website. So if you come up with a new library and you want to publish it and make it available for the community, please contact us. In the site you can also find the contact. And so where do you find all of these libraries? Is in the libraries site, and also you can find there the emojis collection, which is growing constantly. And if you have any ideas for new emojis, I would love to make them. I love when people use them, and that's it. I will release it to the R&D session.